అసలు పిల్లలే లేరు కదా సార్.
Very good afternoon, my dear friends. So today's resource person, Dr. A. Shailaja Madam Garu, hearty Madam. Madam is going to explain one of the important area in plant anatomy. So my dear friends, listen carefully. Please, start your session because students will join during our session. Now please start your screen.
Okay, sir. Thank you. So in the yesterday's class, sclerates, types of sclerates completed, but remaining part, that is the economic importance of fibers. So in these some important examples are there for the fibers. So that the cotton fibers example is a Gossypium, the jute example Carchorus in dicots, Ramiae and Boimeria, agave, Musa textilis and in monocots. So these fibers which occur as bundle sheath are also considered as fibers. Very, very important, these examples. And some hard fibers and soft fibers are also there. These are hard fibers. Examples are we are very seen in that in common in monocots. Example agave and Tillandsia, Musa textilis. These are hard fibers, and soft fibers. Soft fibers, their cell walls. That's why we are called soft fibers. Soft fibers examples are flax, Ramiae, and jute, Madras hemp, kapok. So flax, linum, Ramiae, Bamiria, and jute, Carchorus capsularis. Madras hemp is the Hibiscus cannabinus. Kapok is the Seba pentandra. These soft fibers, these are called soft fibers. It's very, very important. After we are
Madam, your screen is not visible, madam.
Okay, sir. So now I will start the complex tissues.
Okay, madam.
Sir, now is okay? Sir, screen is visible now? Sir, excuse me.
Yes, madam, it is visible. Please you start. Please proceed.
Okay. So simple tissues are completed up to now. So next, we are going to discuss that complex tissues. It's very, very important. In this type, that in the complex tissues, it is a permanent tissue having more than one kind of cells. So in this type, different types of cells perform the different functions in only one system. That is called complex tissues. These are composed of only parenchyma and sclerenchyma without collenchyma. So in this tissue, that xylem and phloem are common complex tissues. All types of cells act as a functional unit and carry on a common main function. All types of cells, different types of cells are present in the complex tissue. Complex tissues are mainly two types. There is a xylem and a phloem. So xylem is the main water conducting tissue in the vascular plants. So that second one is the phloem. It is the food conducting tissue. Xylem is the water conducting tissue. Phloem is the food conducting tissue of the vascular plants. These two tissues are called vascular tissues or conducting tissues. First, we are coming to the xylem. Plants developed the xylem and phloem for conduction. So already we will discuss that xylem is conducting of water and phloem conducting of nutrients. So So in this, that xylem, which is a part of conducting system, appeared for the first time in the pteridophytes. It is a we are observed that first it is appeared for the first time in the pteridophytes. With the progress of time, it has developed as an important or key tissue in the evolution of plants. So that steel is constituted by xylem and phloem. In the steel, we are observed that xylem and phloem are present. So due to the presence of resident thick walls, it can be easily differentiated from the phloem. So xylem and phloem, and between these two, xylem and phloem, that xylem have thick cell walls and resident thick walls, it can be easily identified or differentiated from the phloem. In the stem, the xylem and phloem occur side by side. Xylem characters are extensively used in the classification of plants. So it is a complex tissue. Xylem is a complex tissue. It consists of 4 types of tissues. So see here, on the base of development, the xylem of base of development, it is only 2 types, that is the primary xylem and the secondary xylem. So the primary xylem forms from the procambium of apical meristem. Primary xylem, it is derived from the procambium. First the protoxylem and later on the metaxylem develops. So that first xylem is developed on that is called protoxylem. Later developed xylem is called metaxylem. Okay, protoxylem and metaxylem. That is differentiated into proto and meta. Protoxylem is the first formed part and the metaxylem is formed later. On the basis of arrangement of protoxylem and metaxylem, these xylem again is of 3 types, that is exarch xylem, endarch xylem and mesarch xylem. See here. So in this picture, that first I will show you exarch xylem. Protoxylem is present towards the periphery and metaxylem towards the inner side is called exarch xylem. See here, protoxylem and metaxylem. So first formed part, that is the protoxylem. After later part is later developed xylem is called metaxylem. So see here that protoxylem is present towards the periphery and metaxylem towards the inner side. Okay, that is called exarch xylem. So example, dicot roots and monocot roots. Next, endarch xylem. See here, in this endarch xylem, protoxylem towards the inner side. The metaxylem is present towards the outer side. Is called endarch xylem. See here, that protoxylem is inner side and that metaxylem is present towards the outer side. That is called endarch xylem. Dicot stem and monocot stem is example for the endarch xylem. And the next one, last one is the mesarch xylem. So see here in this diagram, protoxylem is present in middle and metaxylem is present either side of the protoxylem. So that protoxylem is in middle and that metaxylem is present either side of protoxylem. This is known as mesarch. Middle lo untundi annamata. So example is a cycad stem and a selaginella stem. For that mesarch xylem. Next one is the secondary xylem. So this type of xylem is formed from the cambial cylinder. So this is the secondary xylem, is formed from the lateral meristem. Primary xylem is formed from the procambium of apical meristem, but secondary xylem is formed from the lateral meristem or cambial cylinder. The newly formed cells are cut off towards inside and develop into secondary xylem. It consists of different types of elements, that is tracheary elements, consists of tracheids and vessels. and fibers, xylem parenchyma. So in these, we are observed that tracheiry elements that consisting of two types, that is the tracheids and vessels, fibers, xylem parenchyma. Okay. So in this picture, that is a tracheid and vessel pictures. These are the elements. That is a tracheid and a vessel. Okay. Next, we are coming to the tracheids. It is the basic cell type in the xylem. Tracheids are basic cell type in the xylem. These show longitudinal cell walls. The cells are long with a stretched end and appear in the form of a tube. ఇక్కడ చూడండి ఇందాక diagram లో that is tracheid. Okay. So in the first picture you have to see tracheid. This is the cells are very long with stretched ends and appear in the form of a tube-like structure having that tracheid, and the cells are spherical or angular. They are arranged in longitudinal successive rows. That pores are absent in the end walls of the tracheids. Pores are absent in the end walls of the tracheids. This is considered to be an important character of tracheids. The pores are absent in the tracheids. See here, elongated cell with the tapering ends. It consists of lignified wall. It becomes dead at maturity. So it consists of lignified wall. It's very, very important. Tracheids are connected to one another laterally. Bordered pits are found in the cell walls in rows. The tracheids provide mechanical strength to the plants. So it will be give that mechanical strength to the plants. Presence of tracheids is regarded as primitive. These are present in pteridophyte and gymnosperms. It is absent in angiosperms. The tracheids absent in the angiosperms, but in the drimys and tropodendron tracheids are present. So where these two plants, we are observed that tracheids are present in these two plants, that is angiosperm plants, drimys and tropodendron. They are useful in conduction of water and mineral salts. These are the tracheids. It's very, very important. Drimys and tropodendron, that angiosperm plants, generally that tracheids are absent in the angiosperms, but only two plants having the tracheids, that is drimys and tropodendron. Please remember this. And next one is the second one, vessels. These are found in angiosperms, but it is not found in Winteraceae and Trochodendraceae. So it is also important. Vessels are found in angiosperms, but it is not found in Winteraceae and Trochodendraceae. These are known as vessel-less angiosperms. So that two families, Winteraceae and Trochodendraceae, that families are called vessel-less angiosperms. Vessels is a long tubular conducting structure of xylem. Vessels connected end to end. The vessels are dead at maturity. The pores which are present at the end the end walls of vessels are called a perforation plates. See here in the tracheids, already we have discussed just now that pores are absent in the tracheids. That is the differentiation between the tracheids and vessels. So in those vessels, the pores are which are present at the end walls of the vessels. These are called perforation plates. That is important thing. And they are simple perforation plates having only one large pore, and compound perforation plates having many pores. So in that simple perforation means only one large pore, and the compound perforation means that place having many pores. In the vessel elements, secondary wall is formed over the primary pores. The first formed wall is known as primary wall, and later another wall is formed over the primary wall, and this is called secondary wall. Okay, first formed one is called primary, and after that lateral is called secondary wall. The secondary wall exhibits different types of depositions They are, see here, this like annular thickenings, spiral thickenings, scalariform, and reticulate, or different types of vessels are there. That is annular thickenings. Wherever in the annular thickenings, see here, secondary wall thickenings formed as rings on the primary wall on the vessels. Annular means rings like structures. That you have to see that first picture A. Okay, you are observed that that secondary wall thickenings formed as rings, ring like structures on the vessel. Okay, that is the annular thickenings. Next, spiral thickenings. It is the secondary wall is formed in form of a spiral on the primary wall. Spiral means spring-like structure, that is in B. In picture B, you have to observe that spiral thickenings. And after that, third one is the scalariform thickenings. Secondary walls are formed as transverse bonds in the ladder. That ladder type, that is, you have to see in the picture C. You are observed that secondary walls are formed as transverse bonds. We can observe that transverse bonds between that secondary wall. So that is the scalariform thickenings. And after that, reticulate, secondary walls are formed in the form of a net-like structures, reticulate, jalakara antam kada. So secondary walls are formed in the form of a net. So in the D and F, so D, that form of a net-like structure, that is we are called the reticulate thickenings. So the scalariform fitting example is Pteridinium, magnolia, and vitis. Scalariform fitting, that is examples for that Pteridinium, magnolia, and vitis. Next one, we are coming to the xylem fibers. Third one is the xylem fibers. So tracheids, vessels, and after that third one is the xylem fibers. Fibers associated with the xylem are called xylem fibers. So these are long, elongated with the tapering ends. The cell wall is made up of lignin. Sir? My voice is clear? Any? Hello? Okay. These xylem fibers are long, elongated with the tapering ends. The cell wall is made up of lignin. These are dead cells. Xylem fibers are dead cells. They provide mechanical strength to the plants. These fibers are two types mainly. That is, they are libriform fibers have simple pits, and the tracheoform fibers having the bordered pits. Libriform fibers and tracheoform fibers. Next one is the xylem parenchyma. Fourth one is the xylem parenchyma. So this is the only living tissue in the xylem. All are having the dead cells, but parenchyma. So that's why this is the living tissue. The cell wall is made up of cellulose. These are helpful in storage of food material. The rays in them are known as xylem rays. That may be axial or vertical or ray parenchyma. So different types of parenchyma is there. These are made up of parenchyma. They help in lateral conduction. They help in lateral conduction. So out of these, what are the differences between the tracheids and vessels? I will tell you that tracheids are very short, vessels are long. The length of the tracheids is roughly up to 1 mm, sometimes up to 1.2 mm, but vessels length is up to 10 centimeters. Rarely it may be 2 to 6 meters. Example, eucalyptus. So in the eucalyptus, we are observed that the vessels may be 2 to 6 meters. The tracheids, single cells with stretched margins, but in the vessels we are see that cells are arranged one above the other. Okay, these are the differences between the tracheids and vessels. This is the xylem. Okay. Next. Next is the phloem. See here. Second one is the phloem. So xylem and phloem, these two belongs to complex tissues. It's very important. Phloem is also, it is a food conducting tissue. Xylem is a water conducting tissue. So in the xylem, we are observed that the tracheids, vessels, xylem fibers and xylem parenchyma, 4 types are there. Next, phloem. The phloem tissue, it occurs throughout of the plant body. The phloem tissue on the base of its origin, it is of 2 types. There is a primary xylem and primary phloem and a secondary phloem. Same, and xylem also that can be divided based of its origin, 2 types, primary xylem and secondary xylem. So this phloem also same. Primary phloem, this phloem develops from the procambium. Same in the primary phloem, the first formed phloem is known as protophlloem and metaphlloem. Same in the xylem also, that first formed xylem is called protoxylem and latter one is called metaxylem. Same that phloem also same, which develops after or latter, that stage is called metaphlloem. In protophlloem or narrow, in metaphlloem, the sieve elements are short and wide. Okay, so in the protophlloem, we are observed that narrow sieve elements are there. In the metaphlloem, that short and wide elements we can observe that metaphlloem. So secondary phloem develops from the vascular cambium, primary phloem from the procambium. So after the secondary growth, these secondary phloem we can observe. The rays are present in secondary phloem and it helps in lateral conduction of food. So what are the components in the phloem? See here, the phloem is a complex tissue. In this picture, you can observe that sieve plate, sieve tube, phloem parenchyma, companion cell. So the different type of elements are present in this phloem. Sieve elements, companion cells, phloem fibers and phloem parenchyma. Okay, sieve elements, companion cells and phloem fibers, phloem parenchyma. In that xylem, tracheids and vessels, in the phloem, that sieve elements and companion cells are present in the phloem. Okay, next we are going to one by one. Sieve elements are 2 types. They are sieve cells and sieve tubes. First one is the sieve cells. These cells are narrow and long cells with sharp ends. The cells are very narrow and long cells with sharp ends. It has nucleus and of cellulose. The cell wall is made up of cellulose. These are living cells. See here, remember this, it's very important. The cell wall is made up of cellulose. These are living cells. Sieve cells are living cells. Presence of sieve cells, it's a primitive character. Sieve cells is a primitive character. These cells are seen in pteridophyte and gymnosperms. They are useful in transport of food. They are very useful in transport of food. Next one is the sieve tubes. Presence of sieve tubes in an advanced character in plants. So these sieve tubes, having these sieve tubes, that is a very advanced character. These are seen in angiosperms. The cell wall is made up of cellulose, so it is also living tissue. Sieve cells are also living tissue, and the sieve tubes also the living tissue. These sieve tubes are long and are arranged in vertical rows and are connected with each other, which forms a tube. Tube-like structures are formed. That's why we are called sieve tubes. The cross walls between these cells are perforated in a sieve-like manner. Such cross walls are called sieve plates. So in the diagram, we are observed that sieve plates. The perforation in the sieve plate are called sieve pores. The sieve tube elements are enucleate. These cells have thin cellulose cell wall and vacuolated cytoplasm. Cytoplasmic strands extended between sieve tubes cells through the sieve pores. So see here, these connecting strands are surrounded by callow cylinders in the sieve pores. So this very important in the sieve tubes are the connecting strands are surrounded by callow cylinders. Callow slugs, we are called that callows. They help in the food conduction to other parts of the plant. That is the sieve tubes. And the third one are the companion cells. The parenchyma cells associated with the sieve elements are known as companion cells. Okay, that cells are associated with the sieve elements. That sieve elements means what? Sieve cells, sieve tubes. The companion are long, narrow, having thin walls. The cell wall is made up of cellulose. These are also living cells. Each cell has a big nucleus. Companion cells present in angiosperms only. Okay, see here, this very important sentence. See here, companion cells are absent in pteridophytes, gymnosperms. These are present only in the angiosperms, absent in pteridophytes and gymnosperms. Instead of albumin cells are present. Instead of that, albumin cells are present in the gymnosperms. Albumin cells and companion cells stores the protein. Okay. This is the companion cells. These cells are present in only angiosperms, absent in pteridophytes and gymnosperms. It's very important. And next is the phloem fibers. So the phloem fibers are also known as bast fibers. Bast fibers. Fibers are common in the phloem. These are elongated cells with usually lignified walls. These are dead cells. So wherever you observe that lignified cells, cell walls with lignin, that cells are almost dead cells. Okay, that which cells are having the cellulose, that are living cells. Okay, these cells are dead cells. They give mechanical strength to the plants. That is the phloem fibers. Next one is the phloem parenchyma. Parenchyma present in the phloem is called phloem parenchyma. So in the phloem, we are observed that sieve elements. That sieve elements are two types: sieve tubes, sieve cells, and sieve tubes, and companion cells. And after that, phloem parenchyma. Parenchyma present in the phloem is called phloem parenchyma. These are living cells, and cell wall is made up of cellulose. They stored starch, lignin, and resin. These phloem parenchyma are not present in the monocots. It's seen in only dicots only. This is a phloem parenchyma. Okay, thank you.
Madam, excellent. Thank you very much, madam. Okay, my dear friends, regarding on today's topic, you have any doubts? Madam ji, remaining topic we'll discuss tomorrow, madam.
Okay, sir. Okay.
Okay, my dear friends, have you any doubts regarding of today's topic? Okay, madam, thank you.
Okay, sir. Thank you. Thank you very much.
Really wonderful session.